All right, it's 2, so we'll get started. So last reminders, test 3, Monday. This stuff is on test 4, so no need to worry about all these calculations. Friday, not Friday, Friday, the 16th, no live class. I'll have the video up by 2 if you like to watch it at 2, so you can continue your routine. Then the Wednesday, the 14th, you'll have 2 homework assignments due then. But for Monday the 12th, so this Monday, you'll have no homework assignment. So the one that usually would be due on Monday, I just moved it to Wednesday. So just be aware of that. With that, though, let's jump right into it. And we're going to continue to talk about the plasma membrane of cells. And now we're going to talk about the thermal dynamics of the plasma membrane. And when we're talking about thermodynamics of the membrane, we're talking about substances going through the membrane, either diffusing out of the cell or into the cell. And when you look at this chapter, this section in your textbook, they go over the whole proof and why everything is the way it is. So if you really like math, I would suggest you check that out. You might get a kick out of why things are defined the way they are. If you just want to use equations and you don't really care how those equations came to be, that's fine too. And that's kind of where we're at with this. So that's what we're going to work on to begin with, is just how to use this equation. And let's look at this. So the free energy of transport, remember delta G is free energy of molecule A, is equal to gas constant, temperature in Kelvin, multiplied by the natural log of what is the concentration of A where it finally ends up, divided by the concentration of A where it came from, plus Z, that is the charge of A. So if molecule A, for example, was sodium, sodium has a +1 charge, so Z would be equal to 1. While if it was chlorine, chlorine has a negative charge, so that's equal to -1. So that's what Z is, and this is actually supposed to be Z of A. That A is supposed to be lowercase. Multiplied by Faraday's constant, 96,485, multiplied by this, delta phi, or psi actually. And what this is, it's the electronic potential of the membrane, right? This is the electronic component, because when you are moving charged molecules, they really care if you're moving them into negative or positive space. What I mean by that is, let's say you have a sodium, right, positively charged, and you try to move that to an area that has a lot of positive charge already. Sodium doesn't want to go there because like charges repel. While if instead I have that sodium and I try to move it next to a lot of chlorines, sodium's going to love that because they are opposite charges. So this equation really has 2 parts. So it has free energy due to concentration, and it has free energy due to charge. And we're just adding those together to see what the overall free energy of transport is. So we're going to start today with a calculation so we can get used to how we use this equation. So inside our cells, or in the membrane of our cells, we have sodium-potassium pumps. And what a sodium-potassium pump does is that it'll bring 2 potassium ions into the cell, and it'll pump 3 sodium ions out of the cell. All right, so I'm going to help you with this to begin with. And when doing word problems, I really like to draw pictures, right? So we have the cell, right? And I'm pumping 2 potassiums in. So 2 potassiums go in and 3 potassiums go out. Oops. Or 3 sodiums go out rather. So that's what this pump is doing, right? Now, the membrane potential of the cell is minus 100 millivolts inside negative. Okay, what that means is that from going outside to inside, the difference is negative 100. But if you were going the other way, let's say you started outside, or sorry, you started inside and you went outside, the difference there is a positive 100. That's what the inside negative means. It's telling you what direction is that negative charge. If I'm going inside, the change is negative. But if I'm doing the opposite, if I'm leaving the inside, then it's positive because I say the inside is negative. All right. Now, let's continue to read this. So the extracellular fluid, that's outside the cell, has a concentration of sodium that is 140 millimolar and 5 millimolar potassium. In the cytosol, so the inside of the cell, is 10 millimolar sodium and it is 100 millimolar potassium. I want to know what the delta G of transport is for 2 moles of potassium going into the cell while pumping out 3 moles of sodium. I want to know, is this spontaneous? And I give you temperature and I give you the gas constant here. Here is our free energy of transport equation, and here's Faraday's constant. See if you can get at least some way through this equation. Try to at least like get different components of it, like try to label variables, see how far you can get with this to get comfortable, or at least a little more comfortable with this equation. So I'm going to give everybody a few minutes to do this. If you have questions, not sure, do let me know. Otherwise, I'm going to let you get at it and attempt this math problem. All right, so you probably don't have it solved quite yet, and you might have got stuck there a little bit, and that's fine. This is kind of complicated because there's a lot of things to think about with this equation, because we're moving two different ions at the same time. So let's break this down one by one. First off, we have to recognize that we're going to do this calculation twice. We have to do the calculation for the transportation of sodium, and we have to do the transportation for potassium. All right, so let's do the sodium one first. So the free energy for the movement of sodium in this is R, 8.314 joules mole K, multiplied by T. T has to be in Kelvin because our gas constant is in Kelvin. So hopefully you remember to go from Celsius to Kelvin. You just add 273. So this is 310 Kelvin. So 8.314 times 310. Multiplied by the natural log final concentration. That's why I did this picture. The final concentration when we're going outside the cell of sodium is 140. Divided by the initial concentration. Well, where it was coming from, the concentration was 10. So that's the concentration portion of this, plus the charge of sodium, so 1, times Faraday's constant, 96,485, multiplied by the change in membrane potential. Well, we started in the inside, which was negative, and then we're leaving the cell. So our change is a positive value, and it's actually positive 0.1. Let's take a look at why I decided on 0.1. Right, the inside of the cell is in millivolts, and Faraday's constant is in volts. Right, so always be aware of the units that you're using. So I have to convert my millivolts to volts. So 100 millivolts for every 1,000 millivolts, that's 1 volt. So that's why it's 0.1. So before I move on, is there any questions on how I decided on my variables for the free energy and movement of sodium? Now, one other tricky thing about this equation is that we are moving 3 sodium ions, not just 1. So we have to do this equation 3 times, or that basically means we take this whole thing and multiply by 3, because each ion is feeling this free energy change. Or it's actually 3 moles, because I'm wondering 3 moles of sodium. So I have to multiply this by 3 moles. When you do all that, assuming I put everything into Excel correctly, the free energy for movement of sodium is 49,360 joules. So that's how many joules it takes to move 3 moles of sodium outside the cell based on these conditions. Let's move on to potassium then. Same idea. R and T are the same. Natural log, concentration final. The final concentration of sodium is 100 divided by initial, which was 5, plus the charge of potassium, which is 1, times Faraday's constant, 96,485, times here, we're going inside the cell, so it's actually negative 0.1 instead of positive, because we're going the opposite way, And just like the sodium, for potassium, I want to know I want to know the energy for 2 moles. So I multiply this by 2 then. And let me do that. And when you do that, when you multiply everything by 2, I got the delta G for potassium is negative 3847 joules. And I'm just going to double check my numbers here. Yep. And then finally, since this process is going on at the same time, we need to add the free energy of movement of sodium to the free energy of potassium. So I'm just going to add these 2 delta G's together. So it's delta G Na plus delta G K to get total energy of movement, which is 45,513 joules. And hopefully you remember, if a free energy is positive, that means it's non-spontaneous. That means we have to put energy in. And for the sodium potassium pump, one way we can get energy is through ATP. So we can use some ATP to power this non-spontaneous reaction. But yeah, that is the thermodynamics of moving ions across the membrane. You have to take into consideration concentration on both sides of the cell, the charge of the ion, and the membrane potential on each side of the cell. So with that all laid out there, is there any questions about anything I did here? Why did you use extracellular fluid as numerator in sodium, but in K you use cytosol as numerator? Because it's final divided by initial. It's basically where you're ending up, the concentration of where you're ending up, divided by the concentration of where you initially began. So the sodium is leaving the cell, right? So the final location is the extracellular fluid. The potassium is going inside the cell. So the final concentration is intercellular, the cytosol. They're going opposite ways. So what's final in one direction is the initial in the other direction. It all depends on which way they're going on what's the numerator and what's the denominator. That's why I have final and initial, final location divided by initial location. All right, there's no other questions. We can move on. And I believe, yep, that's the end of the PowerPoint from Wednesday. So let's jump into the one from for today. And we are continuing to talk about the membrane and movement across the membrane. And we're going to kind of pick up where that last question left off and look at the types of ways we can move ions across the membrane. Ions are charged, and so normally they cannot go through the membrane by themselves because remember, you have lipid tails here, hydrophobic, and so no ion wants to be in a hydrophobic environment. They cannot have bonds really with hydrophobic molecules. So we have different ways to increase the permeability of ions in the membrane, and in general, they're called ionophores. And there's two different types. You have a carrier and a channel, and they are exactly what they sound like. As shown in this picture, the carrier will bind your ion, and together as a complex, they will just diffuse through the membrane and deposit the ion on the other side of the membrane, while the channel forms a little channel that the ion can go right through. However, these ionophores are extremely specific in that they only allow one type of ion, and they are made for one type of ion. As shown here, for the transporter of potassium, which is made out of these amino acids, the potassium ion is coordinated, it's interacting with a lot of negative charge, and potassium is the perfect size to interact with all of these atoms. If a bigger molecule tries to go through, they will have steric clashes. They'll bump into all these oxygens and won't be able to go through the channel. If a smaller ion tries to go through, then they're too small to interact with all of these oxygens, and they won't want to go through because they're not interacting with anything in the channel. So they're going to stay in solution. While potassium is, it's the perfect size, right? Just like Goldilocks, they're a perfect fit for each other, just right, and so it can go through the transporter. So every type of like ion transport is really specific to try and transport only one thing, one type of molecule. So just to make sure we understand the chemical nature of what does and what doesn't need a transporter, here are 4 molecules, and I just want you to try and figure out, do you think these molecules need an ionophore to go through the membrane? Or can they just diffuse? So they need non-mediated diffusion or mediated transport. So give like 30 seconds to a minute just to look at these molecules and figure out, do you think they can just diffuse or not diffuse? All right, so we're going to start with the actual hardest molecule, which is shown here, ethanol. Because even though ethanol has this alcohol group, which is polar, it's actually small enough and non-polar enough that it can go through the membrane. So ethanol can go through, while glycine needs mediated transport. So ethanol non-mediated. Glycine definitely mediated, because although it's the like same size as ethanol, it is charged, and charged molecules do not go through the membrane without assistance. Don't care what size you are, you're not going through the membrane. Cholesterol is big and has a little OH group, but it's mostly nonpolar. That's non-mediated. cholesterol could go right through the membrane, doesn't need a transporter, while something like ATP, very negatively charged. So ATP definitely would have to be mediated, although I'm not sure why you would dump ATP out of the cell to begin with. But if you did want to do that, you need some kind of transporter to do that because it's not going to diffuse. So any questions about what type of molecules would or would not need mediated transport? All right, let's move on then. So let's look at some of these ion-4s in more detail, just to see how specific they are for the molecule they are bringing across. So first, we're going to look at multiporins. And a multiporin is a beta barrel membrane protein. So you can see the beta sheets forming the barrel. And what they are doing is they're bringing sugar molecules across the cell membrane. So one important class of these are your glycoporins, which bring glucose into the cell, as glucose is the energy of cells. Here, we're looking at the multiporin, which brings in maltodextrins, which is just maltose sugar. And if we look at this, it's kind of like an hourglass shape opening where you're big up here. Let me get a white pen. You're not going to be able to see that black. Where the opening is big, and then it shrinks, and then opens up again. So that is kind of what the channel looks like. And inside the channel, on the very edges, all these yellow things are your aromatic side chains. And aromatic, if you remember from organic chemistry, just means carbon rings. So you have your tyrosines, your tryptophans, and your phenylalanines. And the way that this brings in our sugars is that, if you remember from the sugar chapter, which I hope you do, because we have a test coming up on Monday that sugars are a part of, these sugars are flat rings themselves. Well, not flat rings, but they're rings. And they can do base stacking. Again, from organic chemistry, if you have two carbon rings on top of each other, that's a favorable interaction. And it's called base stacking. So the glucose from our maltodextrins will just base stack with these aromatic residues. And you also have hydrogen bonds with all the oxygens of your sugar with different amino acids in here. So you're making sure that throughout the transportation system, this maltodextrin is constantly in contact with amino acids that react favorably. And when you're going through this, you're kind of going through like a screw. You're kind of getting passed through like that. That makes sure that only maltodextrins will go through, right? You have to have all these interactions, and you have to be a specific size, and you need flexibility to go through this chamber, and only maltodextrins can check all those boxes. So something like a potassium ion can't go through here because they would have to interact with all the aromatic groups, and they don't want to do that, right? So only maltodextrins, and that's how a multiporin has been made specifically to let this one molecule go through. So any questions about the multiporin here? right? Now, another channel for small ions is our potassium channel, which will transport, as you guessed, potassium. You have a lot of different potassium channels, but they are basically all the same. That is, they have very similar sequence and they work basically the same. And they are made just like our multiporin to only allow one type of molecule through it, and that's our potassium. So let me get my black pen back out. Actually, I want to start on white pen. Okay, so the way this channel works, if you focus on the image on the left, you can see that we have these 4 atoms right here. We basically stripped away most of the molecule, and we're just looking at the pore itself. And so you have a 4-fold axis of symmetry. So it's just like copied 4 times to make symmetry there. And you have a lot of negative charge in this channel. That's what all the red means. That's oxygens, right? And the green here is potassium. So when the potassium is going through, they're always going to be interacting with these oxygen molecules, and only the potassium is the exact right size to interact with them at all times. To make sure that different molecules can't go through, we have a pore that will change its dimension, so nothing big can accidentally slip through because it will shrink. And we have a selectivity filter up here at the top again to try and make sure that no other ion just goes through. And here's another image on your right here of your potassiums going through, and throughout the whole way, they're just going to be passed from one series of oxygens to another until they finally go out at the end. So like I said, if you try to get like a sodium through there, it's not the right size. It's the right charge, but it's too small. If you try to get water through there, the oxygen of water will repel with the oxygens inside the charge, so it won't want to go through there as well. So that's just how we can make sure, you know, only potassiums will travel through this channel, which is very important because the cell needs to make sure that all the ions inside of it are always being balanced. The concentrations don't get too far out of whack. So here's just a question that ties back to amino acids. As always, it's important to remember your amino acids. So if I had a porin and I wanted to bring through phosphate ions, what amino acids do you think would be at the entrance of this porin to attract phosphate ions to go through me? Good little review here. Or I guess another way I can ask this question is, what is the charge of a phosphate ion? Is it positive, negative, neutral? Is it antimatter? What is the charge of a phosphate? No one remembers the charge of a phosphate? No one remembers the charge of ATP, which is adenine triphosphate? Negative. Phosphates are negative, yes. So phosphate, as looks like we need a little reminder, looks like that. And it makes up the backbone of your DNA, right? Sugar, sugar. And so that would become negative. So that's why DNA is so negative, because it's made out of these phosphate bonds, which means that, well, if I wanted to attract these phosphates through this porin, I should probably have positive amino acids, your arginines and your lysines. So hopefully you didn't forget about your amino acids and their structures and all that. 3, what type of non-covalent interactions are critical for the ion specificity? This is just, I'll just go ahead and answer this. This is the ion interaction between the potassium and oxygens inside the channel. So these are going to be ion polar interactions. Generally going to be that type of interaction where you have a polar oxygen interacting with charged potassium, or charged polar interaction. And 4, and I kind of answered this when I was going over the channel, so let's see if you're listening. So the potassium channel we were talking about, the opening is about 6 angstroms. Water is 2.75 angstroms. Why can't water go through this channel? It can fit. What's stopping it? Repelled by oxygens. Yep. So there's actually 2 things, and I didn't mention the other one, but yeah, so a bunch of oxygens inside this channel, and water would usually not want to interact with oxygens. It could somewhat through its hydrogens. However, the oxygens are too far spaced apart that water can't interact with like anything. So if you remember in the hydrophobic effect, the reason why there exists a hydrophobic effect is because water pushes away nonpolar molecules because it can't hydrogen bond with them. Water really wants to be a hydrogen bonding all the time. If it went into this channel, this water probably would not be able to hydrogen bond because everything's too far away. So it's not even going to bother to try. So 2 things. 1, lots of negative charge. So the oxygen in water is not going to be happy. 2, it can't hydrogen bond with anything because it's too far apart. All right. Now let's look at ions moving through membranes that have a very important function to us, and that is the nerve impulse. All right, so I know the bottom of these graphs are cut off, so let me just rewrite what the numbers are. So this is 1, 2, and 3 milliseconds, and that's 1, 2, and 3 milliseconds. All right, so your neurons have a lot of voltage-gated channels. And that's how neurons really talk to each other, is by releasing signals that tell these channels to open up, right? So what happens is that when your neuron gets a signal from another neuron, that's going to start a chain reaction where you have a sodium channel here or an action potential. And what's going to happen is that sodium is going to flow out of the cell. Oh, I'm sorry. I got that backwards. As soon as I said that, I'm like, no, it does not work that way. Excuse me, everybody. The sodium channel will open and sodium will go into the cell because cells are negative on the inside, right? So you have a signal coming into a neuron, opens up a sodium channel here. Sodium flows into the cell. And what you have is called local depolarization that happens because once all the sodium flows in, the inside will start to become positive, and other sodium channels around here will also open as well. When that happens, a neighboring potassium channel that's right next to the sodium, and let me get a different color, that will also open up. And potassiums will flow out of the cell because all this sodium is flowing in. And enough potassium will flow out that the inside will become negative again. And this is called hyperpolarization. Once the potassium flows out, both of these channels are going to close. But any sodium channels that are to the right of my potassium channel will open up now. Potassium will flow in. When that happens, a new potassium channel will open up. I think I said potassium here. This is sodium. A new potassium channel will open up, letting those out, shutting both of these off. Then repeat. Sodium opens up, in, potassium opens up. Out, 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 so on and so forth until you get to the very end of the neuron, and then you send the signal to the next neuron to continue that pathway. That's how you propagate an electrical signal throughout your neurons. If we look at the charts here, we can match up that information to what the chart is saying. So here the action potential is, if you look at the charge of the cell, so here at about 1 millisecond or a little before 1 millisecond, our sodium channels opening up, which means the inside of our cell is getting flooded with sodium and our cell becomes positive. After our sodium channel opens up, our potassium channel opens up. And potassium leaves the cell. And so now our cell becomes extremely negative because we have so much potassium that's being released. And then both of these will shut off. And then the cell will return back to its normal membrane potential. That is how a nerve works. It works with potassium and sodium channels. And let me go back a slide, then back. Let me reset this animation. So nerve comes in, and here's the depolarization going. Like you can see only part of the cell becomes positive, then I release my signal. And the part that's inside is becoming positive, the other part's becoming negative because the inside is becoming positive due to all that sodium rushing in. And then the potassium rushes out to restore that normal balance. So that is how a nerve impulse works. Any questions about that? All right, if not, it's about 2:50, so we'll call it here. Last reminder, for those of you who came a little late, tested on Monday. 2 homeworks are due on Wednesday, so no homework on that Monday. No live lecture next week, Friday. It will be recorded and it'll be up by Wednesday. I'll have to record it. If you have any questions over this weekend about test 3, let me know. Otherwise, good luck. Studied hard. While you're studying, realize that these nerve impulses are happening, making new connections. And I will see you hopefully next week, Wednesday. Take care, everybody.